Good afternoon, everyone. I'm Jim Griffin, and I'm delighted to welcome you to the annual Nicola David Pinedo Lecture. Nicola David Pinedo was a leukemia and transplant patient who passed away in 2017. Her family established this lecture in her memory to address key issues in relevant cancer research. We're tremendously grateful to the David family for their generosity and very pleased to hold this annual lecture, which bears Nicola's name. Today's speaker is David Steensma, who is well known to many of us because of the 11 years he spent on the faculty at HMS and Dana-Farber as a clinical investigator in the leukemia program. Dr. Steensma received his MD degree from the University of Chicago, followed by residency in hematology oncology fellowship training at the Mayo Clinic, rising steadily to the rank of professor of medicine and continuing a very productive research career focused on myelodysplastic syndromes and related diseases. David was recruited to DFCI in 2009, where he continued his very impactful research in MDS, describing new genetic lesions, leading a series of important clinical trials that have changed and improved current treatment protocols for this formerly really refractory disease. His clinical studies led to advances in the use of key drugs such as the TNF inhibitor etanercept, the hypomethylating agent decitabine, and the JAK2 inhibitor ruxolitinib, among others. Relevant to this presentation, Dr. Steensma led the first trial of a drug that modulates RNA splicing, H3B8800. Altogether, he has authored hundreds of important papers on leukemias, myelodysplasia, and myeloproliferative syndromes, and is recognized around the world as an expert in developmental therapeutics in malignancies. In 2019, he was recognized for these accomplishments with the Edward P. Evans Chair in Myelodysplastic Syndrome Research. Last year, David took another big step and left DFCI for Novartis, where he became the global head of hematology, translational clinical oncology. It's really a great pleasure to welcome David back for this lecture entitled Targeting Aberrant Splicing in Hematologic Malignancies. To the audience, please submit your questions via the Q&A app, not the chat, so that that's the way we'll find the questions at the end of the talk. David, welcome back to the Farber, and we look forward to your talk.
Thanks, Jim. I'm so excited to have been invited for this and to see so many colleagues and friends on the participant list. This is really fun, and I only wish this was in person that we were able to do this. So my disclosures are a lot simpler than they used to be. I work for Novartis, but Novartis isn't developing anything in splicing right now, so you can listen to this with a clean conscience. Some of the data I'll present are data that emerged from a collaboration with H3 Biosciences, and all funds from that collaboration went to institutions. Some of the correlative studies for one of the clinical trials were part of a SPORE that Rich Stone and Ben Ebert led, a terrific collaboration. And the Edwards P. Evans Foundation not only funded some of my research and the Evans MDS Center at Dana-Farber, but also a tremendous amount of work in splicing in myeloid malignancies at various institutions. And so I want to recognize that foundation as well. It really was a huge shot in the arm for MDS research and for research into splicing. So I think I'm the first speaker in this series who actually had the honor to know Nicola and to be part of her care. And you know, she was such a remarkable woman. If you can imagine what it would be like to come 5,000 miles from your home to Boston for treatment, and to have 3 teenage and preteen sons at home, and to continue to have to make sure that they're looked after and they've done their homework and practiced their piano. You know, she was an incredibly strong woman, and I was struck by just, her poise and her incredibly positive attitude. After she passed away, her family has continued her legacy. Her legacy, of course, is her family as well, her three boys who are now young men. On the right, you can see her parents, George and Katie David, with the director of the Cypress Cancer Institute. And they had supported construction of this new building in her honor, where research into leukemia and other cancers will take place. And on the left, you can see the love of her life, Clemente Pinedo, her husband, who was always at her side, just such a tremendous support for her. And he and others in the family have been involved in setting up a foundation called the Aurora Foundation to support patients who are getting cancer treatment in Athens and have to come from elsewhere in Greece. So it was just such an honor for me to get to know Nicola and her family. And, I know George Canelos and George David really hit it off as well. Successful, accomplished Greek men of a similar generation. And so that was a special relationship as well. So I wanted to begin by acknowledging that. Now I'd like to take you back in time to 1977. When I was a kid in New York and went to Yankee Stadium for the first time. It was a tough year in New York. It was the year the Bronx was burning, in the words of Howard Cosell. Up here in Boston, there was a local band named Boston, whose first album was getting a lot of airplay. And the Boston Bruins were in the Stanley Cup final, where they got swept by the Montreal Canadiens. And that almost could have happened this year if it weren't for last night's game. Canadians might still win this year. Over in Cambridge at MIT, a young investigator named Phil Sharp was trying to figure out where newly synthesized nuclear RNA goes, why so little of it was making its way from the nucleus into the cytoplasm. And they knew this from tritiated uridine labeled experiments, pulse and pulse chase experiments, that a lot of it was just being degraded rapidly after it was synthesized. So why was so much of it being degraded? And they had figured out with a postdoc in the lab and a technician that there were conditions under which RNA preferentially hybridized with DNA, and there was about a 9-degree difference in conditions favoring that. And so if you incubated the DNA and RNA together and then did electron microscopy right at that temperature, you could potentially see hybrids like this RNA. And the first observation they made was that there was a little bit of RNA that was right off the 5' end of the DNA that didn't seem to be pairing. And with the help of David Baltimore, they submitted that observation to PNAS. And one of the reviewers said, no, you can't stop there. You've got to look into this further. So they did additional experiments, and they were using adenovirus mRNA and focusing on a gene that encodes one of the structural proteins, hexon. And they found these 3 loops that I think you can see in this, what's now become a very famous electron micrograph. And Phil Sharp just couldn't believe what he was observing, and decided that this was a split gene and coined this term splicing. Now down at Cold Spring Harbor, Rich Roberts was doing a very similar experiment. And together, they shared the 1993 Nobel Prize for the discovery of splicing. I want to point out the title of Rich Roberts' article. You can never get away with this today, an amazing sequence arrangement at the 5' ends. That would be like, Margaret Shipp saying, isn't it cool what we found with lymphomas and how they're separated, or Anne Mullally publishing a paper that says, look at what this cool thing that calreticulin does that we found. But anyway, it got in Cell, got a lot of attention, and it turned out that splicing occurs in a huge amount of genes. Walter Gilbert at Harvard the following year coined the term intron for introgenic regions. He speculated why this might be. We still don't actually fully understand why we have introns. They're not very well conserved between species, but they are present from early eukaryotic life onwards. And exons, which were named after expression, were the coding regions. We now know that more than 95% of multi-exonic genes undergo alternative splicing. And a lot of the different transcriptional isoforms that arise that encode unique proteins are either tissue or development stage specific. There are all sorts of different ways in which alternative splicing occurs. Cassette exons, mutually exclusive exons, poison exons, exon Valdez, no, that's alternative 5' splice sites, alternative 3' splice sites, introns that get retained, lots of different things that help contribute to this protein diversity. And a whole quarter of the genome is intronic sequences. Splicing itself is kind of a strange word. My Dutch ancestors, who were fishermen in the North Sea, would have known this word because it is a Dutch word, and it did start in a nautical context. And splicing is the act of taking different strands of a rope and putting them together, and that's how the word was used for hundreds of years. I've tried this with a tool called a FID. It's actually very difficult. In the 20th century, the word started to take on other connotations, splicing in the AV club sense, where you're putting together bits of film, and then grammatically, comma splices. And then really in the last 20 years, largely the genetic meaning has swept the field, and it now only has a fairly narrow usage in the sailing world. Splicing is governed by these consensus sequences that are present at canonical splice donor and acceptor sites. And then there's a polypyrimidine tract that acts as a branch site. The actual mechanism by which the spliceosome binds and the process, the biochemical process of nucleophilic attack from the branch site, the lariat formation and then exclusion of the intron, which is then degraded, is a very carefully orchestrated process. And the first thing that happens is that U1, small nuclear ribonuclear protein, or SNRP, binds at the 5' donor site, and then some factors that are associated with U2 SNRP bind down at the 3' acceptor site. And one of them is U2AF35, also known as U2AF1, which will become important for our story. And then different other factors are recruited, U4, 5, and 6, and then there is this degradation process. There are some variants. There's a minor spliceosome, U11, U12 splicing, and this is actually clinically significant. There are germline mutations in U4 ATAC that result in primordial dwarfism. That very small man, Nelson de la Rosa, who was a good friend of Pedro Martinez and always used to come to the Red Sox games, had this form of dwarfism. U11 mutations are associated with neurodevelopmental disorders, as are some of the transfer RNA missplicing events. So there are other forms of splicing that are important, but we're really going to focus on the canonical splicing, the major spliceosome. And wow, is that thing complex. I mean, there's more than 250 different proteins, one of the most complex macromolecules in the cell. And at each of the different splicing complexes, there are a different set of associated proteins that bind. And it really is remarkable how this has come together. I have to say, I've always felt kind of bad for U3, which doesn't have anything to do with mRNA splicing. You know, we talk about U1, 2, 4, 5, 6 all the time in this context. U3 is involved in, it's a nuclear RNA, and it's involved in ribosomal RNA self-splicing, so it doesn't get to play with the others. One of those weird nomenclatures like, why don't we have a factor 6 in coagulation? Who knows? So there are also various elements, exonic splicing enhancers and intronic splicing enhancers and silencers that are involved in the regulation that are near those splicing sites. And a number of different regulatory proteins like serine, arginine rich proteins, SR proteins, bind to those, and some of those will be important, as we'll see. I got interested in this almost 20 years ago now, when I was a postdoc in Doug Higgs' lab at the Institute of Molecular Medicine in Oxford. And I was studying this weird thing that happens sometimes in myelodysplastic syndromes, where patients develop alpha thalassemia, not congenital, they acquire it. And you can see, if you do supravital stain with new methylene blue, you can see these golf ball cells, and that's precipitated hemoglobin H. And while I was there, we found, using a old-fashioned U133 Affymetrix array, that there was this gene called ATRX that was underexpressed in a number of patients. And we found all kinds of somatic mutations in ATRX that were associated with this particular phenomenon, which are very rare in other sorts of conditions, hematologic. It turns out 50% of pancreatic neuroendocrine tumors also have these mutations. They involve chromatin remodeling, and that's outside the context of what we're talking about today. But ATRX continues to have some important biology. Alternative lengthening of telomeres also, ATRX is involved in that. One of the things I found that was quite frustrating was lots of different patients with ATMDS who had transcripts that had different splicing isoforms, but I couldn't find a corresponding DNA mutation. There wasn't anything in any of the splicing donor and acceptor sites. There was nothing in the regulatory elements, and I spent a lot of time looking, and I just couldn't find it. And when I came back to Mayo Clinic and set up my own little lab, I found a number of other examples of genes that were misspliced in MDS and in AML. One of them was FAS, and the majority of these FAS alternative splicing isoforms just resulted in premature termination codons and nonsense-mediated decay, but some of them were functional. They lost exon 6, and therefore they lost the transmembrane domain. They were secreted, and that affected apoptosis regulation. And there were a number of other transcripts in which I found this. So I started to wonder, is there some core spliceosomal mutation that might be causing this? And this phenomenon of alternative or aberrant splicing has been described in virtually every process that's important for cancer development that you can think of. You know, probably the best known is apoptosis regulation with BCL-XL and BCL-XS, which are really just different splicing isoforms. But HIF-1 alpha has a distinct splicing isoform. There splicing isoforms that regulate the epithelial-mesenchymal transition, that influence cell signaling, DNA damage, metabolism, lots of different processes that are important for cancer. And in the therapeutic world, they can also be important for therapy resistance. For instance, there's a splicing isoform of CD19 with exon 2 loss that commonly is a resistance mechanism to CD19-directed therapies like blinatumomab or CD19 CAR T cells. In the myeloid world that Jim, you know so well with CD33 and Rich Stone, critical in development of gemtuzumab and in its reapproval. There is a CD33 splicing isoform, also loss of exon 2, that confers resistance to gemtuzumab binding and as a potential mechanism of resistance. And this is true also for BRAF and androgen antagonists and prostate cancer. And so it's a not uncommon mechanism of therapeutic resistance. The real shot in the arm for this whole field happened just almost precisely 10 years ago when Yoshida and colleagues in Japan doing a whole exome sequencing project found 4 different genes that encoded splicing factors, all of which were U2 SNRP-associated proteins binding to the 3' end, which were highly recurrent, especially in myeloid malignancies. And these are early events. They're usually, not always, but usually mutually exclusive. Coleman Lindsley resurrected a phoenix from the ashes of the Ammonified trial and was able to look very carefully at secondary type AML arising from a pre-existing therapy or pre-existing myeloid neoplasm, and showed that there is strong enrichment of splicing gene mutations there. And at least in 3 of the 4 genes, not in ZRSR2, where there's tremendous allelic heterogeneity. It really looks like a loss of function situation. With SF3B1, with SRSF2, with U2AF1, there were hotspot mutations that suggested there was altered function. These are not restricted to myeloid neoplasia. They're certainly strongly enriched, especially SF3B1 mutations in MDS with ring sideroblasts, that particular morphology, but they're also seen in other disorders. For instance, SF3B1 is seen in 15 to 20% of patients with CLL. Kathy Wu has done some important work in the function of SF3B1 in CLL. They're also seen occasionally in solid tumors, particularly in bladder, and there's occasionally pancreas and non-small cell lung cancer. The prognosis of these differs across malignancies. In MDS with ring sideroblasts, they tend to be associated with quite indolent disease, whereas in CLL, it tends to be a higher risk factor, so that the context probably matters. Now, trying to understand exactly what these splicing mutations do and how they contribute to neoplasia has been a challenge. Esther Obeng, when she was here working in Ben Ebert's lab, had created an SF3B1 conditional knock-in mouse with the K700 mutation using the MX1 CRE system. And these mice with aging, they don't get leukemia, they don't get ring sideroblasts. Again, splicing not highly conserved across species, so that may be part of the challenge here with doing murine modeling, but they do get anemic and they do get elevated mean cell volume. And when Esther and her colleagues looked to try to really understand this, there was a block in terminal erythroid maturation. As red cells normally mature, they start to acquire glycophorin A expression on the surface. In the end stages, they lose CD71 expression as they move through that orthochromatic normoblast stage and beyond. And there was definitely a differentiation block there. And you could see this morphologically. She was able to show that the spleens were enlarged and enriched with erythroid precursors. But when competitive transplantation experiments were done, these SF3B1 cells actually seemed to have a disadvantage. So how they gave a clonal advantage was really unclear. The MSKCC group, Omar Abdelwahab and Eli Papamanuel, they did a very similar murine modeling and had quite similar results. In the context of lymphoid malignancies, MYC really seems to be a key player in the context of SF3B1 mutations. And although these SF3B1 hotspot mutations activate hundreds of cryptic 3' splice sites and result in diverse sorts of changes across the transcriptome, probably what's most relevant to lymphoid malignancies is the decay of a phosphatase, protein phosphatase, 2R5A. And this increases MYC phosphorylation and stability. And so you can see when you cross a SF3B1 conditionally mutant mouse with a MYC mouse that they have a reduced survival, they have cytopenias, and they tend to get lymphocyte malignancies with aging. One of the more interesting observations came from Omar Abdelwahab's lab. This was in Nature about 1.5 years ago now. And it really focused on BRD9, bromodomain 9 missplicing. And what they did was to do some comparison of expression profiling in MDS and uveal melanoma and CLL compared with controls. And they looked for genes for which mutant SF3B1 promotes nonsense mediated decay in all 3 malignancies. And they found 7 of them. And one of them was BRD9, which was of interest because they also did a positive selection CRISPR screen, and it came out of that as well. And so when they did RNA sequencing, found this poison exon that was predicted to result in premature termination codon. So what happens with SF3B1 mutations is you get this exon 14A, which gets included in the transcript, and then there's a reduction in BRD9 that influences a non-canonical BAF complex, which has a significant effect on expression of a number of different genes, and results in the degradation of the BRD9 transcript and a net loss of functional BRD9. So really, I think an important observation, whether this is, the missing link between all of the different SF3B1 isoforms, I think we don't know. Well, I'm a clinician and a drug developer, and that's certainly what I fill my days with now. So what was most of interest to me was, what can we do about this? Now that this observation is out there, that there have been tremendous increase in the understanding of the biology, what can we actually do with malignant splicing that might help our patients? Omar published a review in Nature Medicine about 5 years ago, speculating on different ways in which this splicing might create a vulnerability, a sort of non-oncogene vulnerability that could lead to something that could be targeted clinically. The first and most obvious would be to use agents that actually bind to SF3B1, and by further inhibiting its function, you could cause synthetic lethality, you could cause selective killing of the clonal cells, thereby sparing the healthy cells that have residual intact splicing. Some of the other ideas were inhibitors of phosphorylation of those SR proteins that I mentioned earlier that bind to exonic splicing enhancers and silencers. Potentially, if these splicing isoforms were present in enough patients, you could design an immunotherapy that's specific for an abnormal splicing isoform, and that could be clinically useful as well. That's actually been really quite hard to do, to come up with something that targets aberrant protein product that's present in enough patients for it to be viable targeting. And then there's some other targets that weren't thought about really at the time Omar did these reviews that I'll talk about at the end. PRMT5, protein arginine and methyltransferase, which is involved in methylation and assembling of these SNRPs. RNA binding proteins, I'll mention RBM39, and then ATR, which is a local project where Andy Brunner and Tim Graubert over at MGH have played a leading role. And at the Farber, we had enrolled patients as well. And ATR is activated during our loop formation during splicing being co-transcriptional, and that creates a vulnerability as well. Now, there had been clinical trials of a compound called E7107. It's a platinumide derivative, a natural product that was tested at MD Anderson in solid tumors because it was a cell cycle inhibitor. They didn't really understand the splicing aspect of it at that time. And unfortunately, development was suspended because of toxicity, ophthalmic toxicity, loss of vision. Which probably was an on-target effect because there is a form of autosomal dominant retinitis pigmentosa that is due to a germline mutation in PRPF40, a minor splicing factor. Relatively recently, CLK inhibitor and the first SRPK inhibitor have entered the clinic, one in colorectal cancer, one in sort of a general solid tumor model. Phase 1 trial system, and there's no clinical data on these yet, but these are other ways. The others on this list, spliceostatins, herboxidines, these are really tool compounds and not useful for clinical use. So Ben and Esther had access to E7107 and put this in their murine model and did observe some selective cell killing with it. So this is where H3 Biosciences got involved, and they had have a program, they're still a going concern, focused on modifying the groups around this heterocyclic lactone ring to achieve better cell activity against SF3B1. And the compound that made it into the clinic was 8800. And what that does is binds to a pocket, which binds to the mRNA. And that pocket's created by the binding of PHF5A and SF3B1. And there's an ATPase there. The structural chemistry has been very nicely worked out, as well as potential mechanisms of resistance that close this pocket or alter the conformation. So Omar Abdelwahab did some of the preclinical work with 8800 in a CMML PDX model, P95, which is one of the hotspots on SRSF2. And as you can see up at the top, there is a population of cells that is present there in the vehicle, in marrow, spleen, liver, et cetera, but goes away with treatment with H3B8800. On the bottom with splicing wild type, there isn't that selectivity, suggesting that not only is there selective killing of splicing mutant cells by this additional spliceosome stress, but that a healthy cell, a normal cell, can tolerate to some degree inhibition of splicing or modulation of splicing. So we did a clinical trial, and this trial took a long time to accrue to because we had to go through a lot of different dose levels. It was for patients with MDS, AML, or CMML who met specific entry criteria, including having nearly normal vision or normal corrected, because we were very concerned about the ophthalmic safety. That turned out not to be an issue, thankfully, probably because in part we built in such a careful monitoring program. Based on the Sino-monkey data, we started out with a 5-day on, 9-day off schedule, but weren't seeing the clinical results that we wanted. So introduced a second schedule with 21 days on and 7 days off. And this is the classic table 1 showing who the patients were who went into this study. It was a roughly equal mix of MDS and AML. The majority of patients did have splicing mutations. We actually didn't require it because we wanted to know what the compound might do in non-splicing mutation patients as well. But of course, clinicians select who goes on clinical studies, and all of us who are enrolling to this trial picked patients who had specific splicing mutations. Many of them were pretty heavily treated. It was an older population group, median age of 74, and almost all had had prior hypomethylating agents and were receiving red cells, and about a third were receiving platelets regularly. The PK was pretty linear as expected, dose-dependent increase. The one thing I think I wish we had done differently was to recognize how quickly this decayed, and that by 12 to 16 hours, there wasn't much of this splicing inhibitor left over, because that might have led us to do a little bit of a different study or open up a different arm. Most of the adverse events were gastrointestinal in nature, which was also what was seen in the cyno-monkeys preclinically. We thought, based on local investigator assessment, that there were some QTC prolongations, and that was declared to be the DLT after we'd gone through about 9 different dose levels, up to a dose of 40 milligrams a day. And so the trial was paused. It was also paused in part because we weren't seeing the type of clinical activity that we wanted to see. Of those who were red cell transfusion dependent at baseline, about 15% became independent for at least 8 weeks, and one patient achieved platelet transfusion independence for 20-some weeks. We took a careful look at those patients, and there didn't seem to be any clinical characteristic that define them. And 2 of them are not included in this table here because when we went back and really carefully looked at the data, we couldn't find what their level of pre-enrollment red cell transfusion dependence were. They were just marked as getting red cells regularly, and we can't be sure about that. So in the final paper, we annotated that. We did hit splicing, modulate splicing, certainly. H3 had set up a pharmacodynamic assay using K562 and NALM6 cell lines, and they were looking for transcripts that had certain properties, that the expression of the full length and mature mRNA decreased by at least 1.5 logs, that there was either exon skipping or intron retention, and that was expressed in MDS and AML samples. And that most of them were at least preclinically affected by H3B and E7107. So they came up with a list of about 30 different transcripts. And you can see over on the right here, there is a dose-dependent modulation, a decrease in the amount of the mature mRNA transcript and an increase in the amount of incompletely spliced pre-mRNAs. And there's two specific examples over there on the left. None of these things we know have any relevance to MDS or AML pathophysiology, but they were useful markers of splicing. Since then, they've gone back H3 and looked at the red cell transfusion independent patients. And one interesting observation has been that most of them had aberrant splicing of a particular transcript called TMEM14. And I'll show you in a second what this is, but it's involved in heme biosynthesis. And of the subjects that achieved transfusion independence, most of them had quite high ratio of aberrant to canonical junction expression in the context of SF3B1 mutation. So, there was pretty rapid shift in that relative to baseline with treatment that also fairly rapidly returned back to baseline. So what is TMEM14C? Well, it's required for mitochondrial heme metabolism. It's involved in secreting protoporphyrinogen 9 across the inner mitochondrial membrane. And there is interest by a couple of different investigator groups, primarily Sergei Dulatov out in Seattle, in understanding better TMEM14C. There have been a couple other heme biosynthesis associated transcripts that people have linked though to the ring sideroblast phenotype. ABCB7 is one, PPOX is another, and so I don't think we fully understand this this story yet. It is interesting. I'll skip this one here. So what happens next with H3B? Well, there is a proposal afoot to reopen the study and do BID dosing in a subset of patients with SF3B1 mutations, because we don't really understand why there is such a disconnect between Omar's preclinical data and Esther's preclinical data with the E7107 compound and what actually happened clinically, which was not a whole heck of a lot. One possibility is that it's like the deist hypothesis. Splicing mutations are required to get the clonal process started, but then aren't absolutely necessary in most patients to maintain it. Second possibility is we just didn't hit the target hard enough. And that is still very possible that with increasing the dosing that we might have hit it harder, that we might have gotten into a therapeutic window. We had a cardiologist at Mass General who's an expert in QT electrophysiology, independently adjudicate all of the quote unquote QTC prolongations from different places, all outside of DFHCC. And he felt they weren't real, that the QT prolongation was not real and felt like we could have gone to higher doses. So hindsight's 20/20. So RBM39 is interesting. It's a RNA binding protein that is degraded by the E3 ubiquitin ligase system, something that Ben Ebert and Bill Kalin showed was so important in the mechanism of action of lenalidomide in del(5q) MDS and in myeloma. And by using sulfonamides of a certain type, what this group at UT Southwestern called SPLAM, splicing inhibitor sulfonamides, you could augment that interaction and degrade RBM39 and create the same sort of spliceosomal stress that something like H3B created. And so Omar and Ayanna Saifontis, who was a speaker in this Nicola David Pineda series the last time we held it 2 years ago before the pandemic, had published this really interesting data looking at the sensitivity of AML cell lines to RBM39 degradation, and just Nathaniel Gray, Eric Fisher, Jay Bradner, the chemistry group at Dana-Farber, just unbelievably beautiful work. Really worked out the structural characteristics of this binding. And there was this compound E7820 that is a sulfonamide that binds to this pocket and has similar sorts of effects on RBM39 degradation and on this ubiquitin ligase activity as indicilam, and seemed ripe for clinical trial, but somebody had forgotten to renew the import-export certificate on it. So it's been sitting in limbo for about 3 years, but I talked to Omar last week and he tells me it's finally moving forward, getting very close to being used in clinical trials. In the interest of time, I'm not going to go through this in a lot of detail, but Tim Graubert and colleagues over at MGH have been looking at R-loop creation during splicing. And they found that this R-loop creation is associated with a potential sensitivity to inhibition of ATR in MDS. There's a RPA, replication protein A. It's a sensor single-stranded DNA at stalled DNA replication forks. And if you have missplicing, the replication may stall. That then recruits ATR. And if you then inhibit ATR, you induce more DNA damage in cells with splicing mutations than in those wild type. And if you then on top of it put in a splicing inhibitor to further stall that DNA replication, then you can really get a very potent cellular effect that's specific to the U2AF1 mutant cells. So this led to a clinical trial led by Andy Brunner and Rich and Dan and colleagues at Dana-Farber also accrued to this study and had some patients that had some interesting hemoglobin increases. The study is still ongoing, and here you can see the study schema. And so look forward to seeing what the final results of this are. Last thing I wanted to mention, targeting PRMT5, which is important for SRSF2 and associated factor methylation, and is important for assembly of the spliceosome, and leukemia cells, at least preclinically, have been shown to be sensitive to it. There's now 2 different PRMT5 inhibitors in the clinic. Jacqueline Garcia is leading the trial at Dana-Farber, or it might be Marlies Luskin, I can't remember. The first clinical results in solid tumors were presented just this weekend at ASCO with the Pfizer compound, and they went through a couple logs of dose levels, but really didn't see much clinical activity, 2 partial remissions. They did see reduction in a biomarker, SMDA, and that led them to believe that they were hitting the target. So watch this space. So as of about 2 weeks ago, the H3B trial is finally accepted at leukemia. And so I want to thank all the collaborators on that. You can see them listed there. Rich has really carried on the mantle with this study since I went over to develop drugs in a different way. We cycled through a lot of different medical monitors and staff at H3B. I have to say, it was, for a while, it was like Harry Potter and the Defense Against the Dark Arts teachers. We were getting a new one every year or more frequently. But Antonio Gualberto has been terrific in getting this across the finish line. And then last, gosh, I miss you guys. You know, there are certainly a lot of wonderful things about what I'm doing now. I love it, the science and the scope of what I'm doing. But you know, having the sort of banter with friends and colleagues, with my friends in the DFCI leukemia group, with scientific collaborators, you know, how much have I learned from Ben and Coleman over the years? You know, I can't even count it. So just wanted to acknowledge them. And thank you again to Nicola's family, who have contributed to make this lecture series possible and to spread scientific knowledge that's going to help patients with leukemia and other conditions. And so you're doing in many different ways, carrying on her legacy. Thanks.
David, that was terrific. And let me remind the audience to put your questions into the Q&A box, not the chat box, and so that David can see them. Let me start off with one, and that is one thing I didn't understand from the results of the H3B drug trial is whether or not any of the known abnormal splicing events were corrected, either partially or completely. Say, for example, in BRD9 or TMEM or any of the many things that are misspliced. Did they improve, and did that correlate with any clinical outcomes at all?
Yeah, so we don't know about BRD9. That certainly would be something of interest to look at. One of the problems was that we didn't see a whole lot of responders. I think if we'd seen a group of patients who had CRs and a group who didn't, that really could have facilitated molecular dissection of trying to understand what happened. And I would say with splicing, it's not an all or none phenomenon. It really is a shift in ratios, usage of an exon versus not usage of an exon, even in a story that seems pretty clean, like that poison exon in BRD9. I mean, you can still detect it in various settings. And so we definitely did see, several log change in mature mRNAs that had been missing that then were able to be fully transcribed, as well as a reduction in some of the what would seem to be aberrant or abnormal splicing isoforms. Another hard thing is that, if something is not necessarily the most common splicing isoform, it still could be very important for cellular function. And, that's why we have probably the diversity that we have in splicing. And it's hard to model across species. Mice splice differently than we do. And, the fact of splicing itself is highly conserved, but not specific splicing isoforms or transcripts.
Yeah, that makes sense. There's a question from Rich Stone in the Q&A box, if you want to open that.
Yeah, I got it open there. I think if the question is about should we have seen ring sideroblast morphology change? It was very few patients that actually had follow-up bone marrows during the time when they seemed like they were free of transfusions. And also, even when they were free of transfusions, it's not like their hemoglobins went to 12.5 or 13. They were 9.5 or 10. And so I think it's possible that if we had a really robust splicing modulator that would see morphology changes, particularly if you ended up getting clonal reduction, which was another thing we didn't see. We looked at the SF3B1 mutant burden by variant allele frequency, and it's semi-quantitative. It's not perfect, but it's pretty good, and really didn't see major changes in that clone. So maybe if we could have gotten rid of the clone, we would have seen normalization in that morphology.
Great, and there's a question from Dan D'Angelo there as well.
Yeah, Dan, thanks. That's a terrific question. And I don't know if Ben is on the line. The question is about how exactly do mutations in the spliceosome cause a clonal advantage? I think in some cases, it's probably because of specific transcriptional isoforms like the BRD9 poison exon I showed that then have a global effect on transcription that leads to, in some way, a clonal advantage. But I don't think we fully understand this. And it may also depend on the specific mutation. Coleman has some really interesting data on the characteristics of U2AF1 S34 versus Q157 in this huge database that required so much effort to build at Dana-Farber, and tying in genomics with pathology and with clinical data. And you know, he and Marlies Luskin and Goyo Abel, I think, are going to be able to mine that and try to understand that a little bit better.
Okay, you could go on to the next questions, David.
Okay, yeah. So Sophia has a question. Hello, Sophia. Thanks for coming. H3B8800 drug toxicity and how it could be overcome. You know, most of what we saw was gastrointestinal, and so I think that you know, there are lots of drugs that are irritating to the GI tract for various reasons, and most of the time with good supportive care we can overcome that. There are drugs in which that's just really an Achilles heel and leads to diminished use. Selinexor, a classic example of a drug that required tremendous supportive care to get patients to tolerate it. I think if we end up actually doing this BID, or I say we, the collective we, it's got to be somebody else now, because you know, Novartis doesn't own H3B, they're on their own now. But if we end up doing this BID dosing study, I think we really have to pay attention to that and try to, you know, make sure that safety is respected. And maybe the eye safety will become an issue if we hit splicing harder as well. So Anne asks, in the patients who've had improvements in red cell counts, is the mechanism understood? So we didn't see clonal shifts, and so that doesn't seem to be how those 11 patients or so achieved red cell transfusion independence. So that's not what's going on. What is going on? You know, is it more effective heme biosynthesis because of less missplicing of TMEM14 or ABCB7 or PPOX or some of the other transcripts that have been associated with spliceosome mutations? I don't know. And then Andy, thanks for joining. Andy's leading that ATR study I mentioned at the end, and he asks, you know, can we lump all of these splicing mutations together, or do we need to divide them more for future studies? I think, you know, one of the reasons that they were lumped together for this study, the H3B study, was because it seemed like it was really taking advantage of spliceosome sickness generally with a synthetic lethality approach. I think now we have to take a step back and have a rethink about that, because different splicing mutations have different consequences. I didn't talk at all about ZRSR2, but that's a whole different kettle of fish from some of the others. And what U2AF1S34 mutations do in Q157 is distinct. You know, and clinically they're distinct as well. We even see a different gender balance in patients in terms of that mutation rate. So.
Just a comment to the audience. If you have further questions, you can put them in the Q&A or send them to Claudia Steele, and we'll make sure they get forwarded to David for answering later on, perhaps. If there are any other questions or comments? If not, David, great talk. Really enjoyable, complicated, and fascinating topic.
Thank you, Jim. Thanks for having me.
Great. Take care. Good luck over there. And
thank you.